Great. Hello, everyone, again to this second session of the DMS Pro workshop series on marine sulfur cycling. Today, we have two speakers who are Jacqueline Stefels and Todd, who are going to talk about DMSP synthesis, pathways, players, and measurement methods. So before that, I will give you a brief introduction about our working group, DMS Pro. So who we are, what we are planning to do. So DMS Pro stands for Developing Resources for the Study of Methylated Sulfur Compound Cycling Processes in the Ocean. So we have a common interest in the organic sulfur cycling in the ocean, in methylated sulfur compounds. And this is the group with about 21 members from, I think, 13 or 14 different countries around the world. You know that SCOR has quite stringent criteria on on the distribution of members across countries and genders and expertise also. Good. So what is this group? What are we planning to do? Here you can see the three main deliverables we would like to create. And in first place, we want to create a standard operating procedures documents, so kind of standardized protocols agreed on by the community. And in fact, this discussion we'll have today is very relevant towards these objectives of writing the SOPs. And second, we are also starting to design and create a database of cycling rates of these compounds, which can then be used for everyone to contrast their own measurements for modelers, for validation and model development, and so on. And finally, we are planning to write some scientific articles describing the database, providing guidelines for data users, and also some perspectives on this research area. So overall, we want to strengthen this community of practice around the research on sulfur compounds. Great. So many or most or all of you are probably familiar with this entangled scheme of the several cycling pathways of the methylated sulfur or methylated sulfur compounds and their related metabolites, some of which are not here, like acrylate. This was from a review by Jacqueline Stefels, our speaker today, back in 2007. And since then, there has been many discoveries concerning new pathways, new compounds, or activities, unexpected activities of some organisms in the plankton. So we know this needs update, and this is one reason for our group to exist, updating the scheme and providing some quantitative understanding for these rates and the capacity to measure all of them in the most standardized way as we can. Another reason for creating this group was the perception that there was a steady decline in the number of publications or the number of studies about these compounds, like where in particular studies where cycling rates were being reported, were being measured. And last spring, we did a literature compilation in DMS Pro. And when analyzing the data, in fact, we could confirm this perception that for the last decade, there has been a decline in this kind of measurements, which in part has been has co-occurred with the advent of or the generalization of molecular techniques. But just because molecular techniques provide new understanding, we should also be measuring rates at a pace that allows us to compare rate activities, actual activities in seawater with molecular insights. And our talks today, in fact, are covering very complementary aspects because in first place, Jacqueline Stefels will talk about phytoplankton DMSP synthesis and the method she developed to measure these synthesis rates. And in second place, Todd is talking about molecular insights into microbial DMSP synthesis and pathways. And you probably know our two speakers. But basically to introduce them very briefly, Jacqueline her expertise has made remarkable contributions on the ecophysiology of phytoplanktonic DMSP production and related activities, and John talked about the molecular aspects of DMSP cycling, but also DMS, acrylate, and so on. So we have these very nice speakers today, and we hope you enjoyed the talk. So I will stop sharing the screen, and the floor is yours, Jacqueline.
Okay, I'll share. Okay. Do you see my presentation?
Yes.
Yes, in the correct mode as well. Okay, good. All right. Well, thank you for inviting me and for organizing this all. So I will say a few words on the mass ratio progress method that I developed indeed in 2009. So the main problem that most people are worried about is the ongoing question that what controls the production of DMSP, and how do DMSP, and that derives from that, how do DMSP producers actually respond to environmental stressors? Now, conventional gas chromatographic methods only measure the net change in the methylated sulfur compounds. As long as they are unable to measure all the sinks because they can't be measured or they are not even known, and cell lysis in incubations is going on, it is very hard to get rate measurements, rate numbers from these experiments with the conventional methods. And I'll show you here that the solution is the C13 incorporation with mass spectrometry. For those who are not at all aware of any physiological experiments, a few slides on how these experiments work. So basically, 13C carbon, so the heavy partner of the natural 12C, is one mass heavier because it has an extra neutron, and because it's only a neutron that makes it heavier, it doesn't change the reactive sphere of the electrons around the nucleus. So it's basically an ideal compound to look at incorporation of carbon. Sorry, I have a bit of a cold over the last week, but I'll try to not to have a normal voice. I'm sorry about this. Anyway, so there might be a bit of a fractionation going on when you offer C13 instead of C12 to your organisms to feed on, but in fact it's even less than when you would offer a radioactive 14 carbon, which is even heavier than the 12C and the 13C. So where does CO2 comes in? So normally photosynthetic organisms have chloroplasts, and they get they convert solar radiation, the solar energy, into chemical energy, and the chemical energy is then used to fix CO2 into sugars. And sugars can then, via many more steps, being converted into DMSP. So this is the basis of the whole system. And CO2 uptake can only be done through autotrophic organisms. So the mass ratio progress method is based on the very simple analysis of DMSP that everybody does. So conversion with NaOH and the measurement of DMS. and I'm not bothering about the other products. Now, DMS has a mass of, in total, 62, which is depicted in this little cartoon down here. But for a proton transfer reaction mass spectrometry, that's the mass spectrometer that I'm using, and which is very efficient in measuring DMS. One proton is added, so in fact, mass 63 is being measured as the moiety for DMS. So in the method further down the line, I will refer to mass 63 as the natural DMS mass. So if you now add CO2 through photosynthesis like this, then part of this 13 carbon is being incorporated into the DMS moiety of DMSP. For sure, it will be incorporated in all carbons everywhere, but we are now only looking at the DMS moiety. And hence, if C13 is incorporated, you get one mass heavier, which is 64 in the PTRMS analysis. Now, the chance that this one carbon within DMS is being converted into 13C is, for instance, if you add 3% of C13 is 3%, but the other carbon can also be, has a chance of 3% to be added. And in addition, of course, there are also possibilities, statistical possibilities, that you have 2 times 13C in your DMS moiety. So you can calculate the maximum chance of one C13 added to your DMS through binomial distribution. And in this case, if you add, that means that if you add 3% of C13, then the chance of finding one mass heavier in your DMS is 5.82%. And in addition, we have natural background of stable isotopes, which I will show in the next slide. So the mass spec measures all these different masses, and also mass 65, which is measured. So what I will mention later on is the ratio of mass 64 over the total in percentages. So I mentioned the natural background because both protons, carbons, and sulfur have natural stable isotopes, and that all adds up to a specific mass fingerprint of DMS, which has, as is shown here, a certain fixed mass in higher masses. Yeah. So how does it work? So if you have, for instance, here a hypothetical growing culture of algae that by definition grow exponentially, and then you add your stable isotope, then the addition of the stable isotope in the pool of DMSP will start with the background ratio, or RB, and will go towards equilibrium ratio, which I call here RM, so the maximum ratio. It's all depicted here. So in the beginning, so what the mass spec measures is basically just the ratio at time t. And at time t, there you still have a fraction of old DMSP that has the background ratio, and you have a fraction of new DMSP that has the maximum maximum ratio, the equilibrium ratio. We already saw that the algae are usually growing, or you can state that that they grow exponentially. And if you combine these two equations, then you can get rid of the pool data, so the N's. That's the concentration of DMSP in your culture, and you're only left with ratios. And with that, you can calculate the rate constant based on only ratios. So, and this gives you the specific growth rate. So it gives you production of DMSP per DMSP present. And therefore it is, the unit is per time, whatever time you are using. And this works so beautiful because you are not you can be sloppy in collecting your DMSP data or your samples. The ratio will always be the same. It doesn't matter whether you spoil half of it and you didn't notice it. The ratio is always the same, and that it works beautifully is shown in this this one example. So, for instance, this was an incubation with natural cultures, and you see that the pool goes somewhere, like just everywhere. Well, everywhere, yeah, but there is not really any trend visible in the pool of DMSP. Whereas the ratios are gradually increasing. And with these ratios, you can calculate for each section of the period how much the growth was. So DMSP synthesis rates can be determined very accurately, since mass ratios are more accurate than concentration measurements. Now, there are some problems with it. Well, problems, you cannot actually solve the problem, but you can imagine that if you do an incubation with a lot of stress, then cells can, for instance, blow up. They can lyse due to high light conditions, or they lyse because of grazing or whatever. And what happens then is that the production starts with works with produces DMSP with the highest ratio, but the loss is mainly from the already present pool, which has the background ratio, and that means that the newly formed DMSP adds quicker to the ratio of the total pool. So that's depicted over here. So for instance, if you do an experiment and basically your DMSP pool goes down because there is more loss than production, then you will find in the mass spec the green ratios, whereas the actual growth of your algae that you want to study is following the brownish ratios and is therefore lower. And I've provided that in the same paper also with this model outcome, which shows that that if you have very low DMSP production rates but relatively high loss rates, then the difference between what you think that you are measuring and what the actual growth production rate is relatively high. So, but of course, that all depends on how long you wait until your T0 and the moment that you get your sample. And as the previous example already showed, if you give if you follow your incubations for only a few hours, then usually that suffices and gives you a proper estimate of the production rates of phytoplankton. So it's a very, so the take-home message from this is that the DMSP synthesis rate can be very determined very accurately in combination with the particular DMSP concentration measurements. And then the old problem comes back again, that you have to do that neatly, and concentration measurements need to be very accurate. Then you can you can get a simple model in combination with the ratio measurements, and then you can in fact solve both the production rates and the loss rates mathematically. But that, I mean, that's too far to go into detail in this, because that's basically the topic of next week's workshop. But it does show that the loss rate is always more variable than the production measurements, calculations, and because the loss is much more complex, and because you need concentrations, measurements, etc. So if so, I wanted to give you a few slides on how we use this method, and what we can learn actually if we in parallel to the DMSP synthesis measurements also measure primary production rates. And so very often the question is how is DMSP regulated in the algae that we want to model, or that are very abundant in the environment, etc. So one of the questions is it actively upregulated under variable environmental stressors, or is it just simply coupled to carbon assimilation? And we use this. We took this question in experiments to look at UV stress. We all know that UV stress gives a lot of photooxidative stress. Since it was shown that DMSP is capable of scavenging oxygen radicals, the question is it actively upregulated by the cell to do that, or is it just coincidence that it can do it? So we did an experiment with highlight and low light levels, and with and without UV radiation. We did that with 4 different algal species, and that were also each had low and high light acclimated subcultures, to see if maybe if you culture organisms for a longer period of time at high light, maybe it adapts its DMSP production and then responds differently. So I'm not going to give you all the details of this paper, because that's a bit too much, but decided to give you at least a snapshot of 2 species. So here you see how the species responded with this method on highlight and low light, and then the overall message is that UV reduced the DMSP production greatly relative to its control at the same PAR level. And that was true for both species. But you also see that low light acclimated cultures had a different response than high light acclimated cultures. That was also similar in these both species. So how does that relate to growth? Because maybe this was just simple growth issue, that they were unable to deal with UV. And then we saw the ratios of, or the production rates of carbon, and that looked almost the same, or maybe not entirely. If you plot here on the right the ratio of the mu DMSP relative to the mu to the growth, that means that if it were to be 1, then there is no up or down regulation. But they were actually different. So maybe there is something. So basically all the low light acclimated cultures in both species were higher than 1, whereas the low light acclimated cultures were less than 1. So what was the reason for it? And then we had a further look into what was actually happening there, and calculated the DMSP produced per carbon being assimilated, because these cultures had adapted their DMSP to carbon ratio in the cell. And then it turned out that there was basically no difference between these acclimations. So DMSP production was simply coupled to carbon assimilation, but there are species-specific differences. And the other two species are also different from these ones. But relative within one species, these data very, very nicely show that it was simply coupled to carbon assimilation. So this is, with that, I want to leave it to that. Thank you for your attention, and if you have any questions, let me know.
Thanks, Jacqueline, for this very nice talk. Maybe we have time for one quick question, and then we move on to the next speaker. And I have to say, I forgot to introduce the schedule for today. So after the 2 talks, there is time for well, and after the questions, we are going to separate like into different breakout rooms to discuss. And after that, there will be a joint discussion, joint debate. So yeah, go ahead with your questions, please. If you have one. I saw one hand raised, or no? So we can leave questions for later, maybe.
I actually wanted to ask a quick question, if possible, to Jacqueline. Thank you so much for the talk. It was super interesting and really well explained. And maybe I wanted to know if basically you could also monitor the with the incorporation of carbon-13, if you could also look at a mass 65 with the PTRMS, if let's say the carbon were incorporated in the two carbons of the DMS.
Yes, that's no problem at all, because it can measure every mass basically from DMS. But as the binomial distribution already shows, it will be very, very tiny. So I do use it because there also is a background ratio, about 4.5% of mass 65, which is already there, which is the sulfur isotopes and other isotopes. And you can see that it increases a tiny little bit, but it is, and therefore I do the calculation of mass 64 relative to 63, 4, and 5, which is the total of everything.
Yeah, that makes sense.
But you see, you see it hardly change.
Thank you.
Well, I have a very quick, well, or maybe not so quick question, but do you think it would be possible to use the same isotope addition to track the losses, the DMSP losses? Yeah, with the same incubation setup, more or less.
Yeah.
Or
Yes.
some of the losses.
Yeah. Yeah. I was first about to show that too, but it was too much. And since losses are the topic of next week's. Actually, I have a few very interesting and very puzzling data on that, because I combined it with the Landry-Hesse, the dilution method, and they give very different results. So I'm not entirely sure how to deal with it, but yeah, you definitely can. But as I said, it is much more, so the data that you get out of it have a much higher standard deviation, because you have to deal with the total amounts, which are always more erratic than the ratios. Yeah, and loss is everywhere. I mean, it can be lysis or grazing or whatever.
That's
So
interesting. Maybe some of this information could go to next week's workshop, perhaps.
yeah, but I don't want to. I mean, there are other people who want to give a presentation, and I mean, I could definitely give another presentation, but that's all non-published and
Yes.
puzzling.
Well, eventually.
Yeah,
Nice to
you
learn
can
more
decide.
about that.
Yeah.
Thank you, Jacqueline. So now we can move on to the next talk by John Todd. So you can share your presentation when you are ready.
Can you see that okay? Yep, you can see that, everybody? Yep, perfect. Okay, so I'm a little bit different from the talkers that we've had so far. I'm a molecular microbiologist, and I agree with the talk so far, in as much as I feel that the process work that we've heard is exceptionally important, and what it should be done is combined with the sort of work that I'm going to tell you about today, which is sort of building on the work that I've done and other people have done, that's looking at the molecular cycling of DMSP production and also cycling. So I haven't comprehensively stuck with what my talk title was, but essentially what I want to give you an idea of is how we can work out what organisms in samples are driving the processes that we're all super interested in. So let's go on to the first slide. Okay, so
John, you might want to go in presentation mode.
I don't know whether that's going to go, and we'll see. If I'll press it and I'll see what it does. If it can you see that okay now, or
no?
Now we see your presenter.
Hello? You see my presenter notes, right? That's what I was worried about. That's why I had it like that.
Okay.
So if it becomes a problem, tell me. So basically,
Well, no.
what we've done over the last hello. So what we've done over the last, I don't know, maybe 15 years or so now, is apply molecular biology to try and understand the pathways and the organisms and the enzymes involved in making DMSP, our favorite molecule down the bottom here, from methionine, but also in the breaking of DMSP to make, of course, DMS and methanephile and such like that. So our work mainly revolves around working on model organisms and identifying the enzymes involved in that allows them to conduct those processes. So what you can see here is the three pathways that are known to generate DMSP from methionine. They're named after the first chemical reaction or enzymatic driven reaction that generates the first intermediate. So we have the methylation pathway, which has been characterized in bacteria, and we've got a paper coming out very soon that identifies the enzymes for DMSP synthesis in Spartina. We find, much like Jacqueline finds as well, we find DMSP in all tested plants that we examine. We identified the key methyltransferase enzyme involved in the transamination pathway. And as yet, unfortunately, we've sort of failed in our trying to understand how the one heterotrophic dinoflagellate takes methionine to make DMSP via this decarboxylation pathway. But the take-home pathway is that we know many of the key enzymes that are involved in these pathways that lead to the production of DMSP from methionine. And another take-home message is that there's huge biodiversity in the ways in which organisms conduct these processes. The exact same can be said for DMSP catabolism, whereby cells either produce the DMSP, they have it intracellularly and they break it down, or organisms import DMSP and they use it as in assimilated processes, which can lead to the production of methane thiol or dimethyl sulfide. So before we started working on DMSP synthesis, we did a lot of work with Andy, who I see is here, which is really nice to see him here. We identified many of the DMSP lyses, of which there are now 10, 9 from heterotrophs, 9 from prokaryotes, and 1 family in algae known as ALMA1. And of course, we have what's believed to be the predominant pathway, the demethylation pathway, that can lead to carbon assimilation, but also sulfur assimilation in the form of methane thiol, predominantly via the first gene, which is, of course, DMDA, or enzyme DMDA. But you can see here again, there's huge biodiversity in these pathways. So trying to work out what organisms drive these pathways we think is incredibly important. And I think it's highly relevant to link to the excellent ways in which you guys are monitoring the process in the environment. So what do I want to tell you about? So the previous talks have nicely summarized how you can study DMSP synthesis and also DMSP catabolism. What I want to show you here is suggest how we can use molecular tools to try and work out what organisms are actually responsible for making that DMSP and or breaking it. So there are a few things we have to get across. The first one is, normally when you guys go out and sample, or when my guys go out and sample, what we typically do is we will fractionate the seawater into bacterioplankton, so between 0.2 and 3 micron size. Or we will take material greater than 3 micron, and we will normally call these bacterioplankton or plankton, or DMSP dissolved and particulate, etc. You then measure the DMSP content in these, or if you're fortunate and you're using some of the techniques that have really nicely been presented, then you can know the DMSP stocks, DMS stocks, but also the rates of production and consumption, which is, for me, a really valuable piece of information to have that we can link the molecular data onto. So much of the work out there at the moment, including stuff from us, is analyzing huge data sets where people have done lots of omics sequencing, but we have no process measurement. So it's really important that all of us sort of interact together and we map on molecular data to the process information that's already out there. So what kind of molecular things can we do? So the first, I've sort of summarized these into different approaches. So the first approach, many of you guys are already doing, it's sort of when you go out and you take your samples and you look for the pigment analysis to suggest what sort of phytoplankton might be in your sample. You also can do microscopy and apportion the ability of some known organisms to actually produce and or cleave DMSP. Some people will, including Steve, who's massive on my screen at the moment, has done flow cytometry to separate the microorganisms by size and by pigments and apportion the DMSP content to those different groups. So all of those techniques in approach 1 rely on you assuming that actually, well, with the exception of flow cytometry, you're assuming that the organisms that you find there are producing DMSP, and you're also assuming the sort of levels that they might be making. Of course, you've got a measurement, but all of these samples are quite complex, and knowing which organisms are mainly responsible is sort of what we're trying to get to. So we'll move on to a completely molecular approach. What you can do and what other people do, probably the easiest molecular approach, is to, when you take your seawater sample and you isolate the DNA from all of the organisms that live in that particular sample. So you filter your seawater onto a filter and you do a standard DNA prep. You can do something known as amplicon sequencing. So the way in which we can taxonomically place organisms is generally by sequencing a conserved gene that's present in all of those organisms. The gene that we typically use for bacteria, and we can use for eukaryotic organisms as well, is the 16S ribosomal RNA gene, which encodes for a protein involved in translation, which is, of course, a universally important process in all organisms. So there are specific primers designed to the hypervariable region, the variable region of that particular gene, which allows you to amplify up that gene and then basically amplicon sequence it. So you get lots of reads from individual 16S sequences that are in that particular sample. And what you can see on the right of my screen now is a sort of consensus output that you'll get from this analysis. What it gives you is the breakdown of the microbial community in your sample. At the top, what you have is we've done 16S amplicon sequencing on DNA, which will tell you what DNA is there. We've also done it on RNA, which we've turned into copy DNA. So this tells us which organisms are likely active in the environment because you're looking at RNA as opposed to DNA. And essentially that gives you a better indication of what the community composition is, and it gives you a more accurate definition of who's there, and applying the knowledge of what we know about these different groups of bacteria and also algae, we can make a prediction of who is the greatest producers, but also cyclers. But of course, that requires a big assumption on what we already know about those different groups of bacteria and algae. What you can see at the bottom is when we actually focus on 16S from plastid sequences, from chloroplasts, etc. So here you can see a study that we did around here in the brackish water samples when we were studying a Primnesium parvum bloom. So you can see that there's a decent abundance of Primnesium here, which of course, as many of you know, can be quite a prestigious producer of DMSP, and it has the ability to cleave DMSP and make DMS as well. But again, from these studies, you can see the abundance of the different organisms that are present in your sample, and you use your knowledge to make a prediction that they are likely key in conducting those processes. It doesn't tell you whether those organisms are actually driving the process in the environment. So if we move on to approach 3, where we're going to use more technical metaomics, if you like. So in approach 2, what we did was we used a technique known as a polymerase chain reaction to amplify a conserved gene. In approach 3, instead of doing any amplification, introducing any bias, what we would do is we would take that DNA, or we would take the messenger RNA, which is transcribed from the genes that are present in the organisms, and we would sequence that DNA and that RNA, which would give us a much orders of magnitude more information about the organisms that are in that sample. This is where it links into the work that what I've done and other people have done on the molecular biology of DMSP production and of course cycling. Because we know key genes that are involved in the process, we can look for the abundance of these genes in that environment, in that environmental sample, and we can apportion the taxonomy from that to tell us which organisms have the genetic potential to conduct that process, but also what organisms are expressing those genes and likely conducting a process. We can go a step further, which I know some people here are actually doing as well, which is even better, which is instead of actually sequencing the transcripts, we can actually isolate the proteins, and we can do mass spec analysis on the proteins that are actually the enzymes that are conducting these processes. So knowing any details on the abundance of the key DMSP liases or DMSP synthesis enzymes and their taxonomy tells us a little bit more information about who's doing it. What metagenomics and metatranscriptomics will also do for you, it will give you much more accurate prediction on the taxonomy of your organisms as well, because you're not only relying on a single gene to work out what that organism is, you're relying on every gene that you've managed to sequence from that organism. Okay, so that's going over approach 3, metagenomics and multiomics. Let's go on to my next slide. So what I want to get across to you is that there are some key limitations when applying these sorts of assumptions to large omic work that we all love to do, and they're very powerful, but they do have limitations that we should all be aware of. The largest limitation, I'm sure all of you have seen papers from myself and also from other people here today, and those who are not here, where we've examined very large data sets where people have gone sampling around the world and have extracted DNA and RNA and have sequenced them to find out what the community is and also what the genetic potential is in that particular environmental sample. Now, what we often do and other people will do is we will look at the abundance of our genes and transcripts in those environmental samples to make a conclusion of who are the major organisms in that environmental sample cycling DMSP, but also those that are actually producing it as well. The best example of this is people analyzing the Tara Oceans data set, which is a massive available resource for people to do, and it makes sense to do it. But unfortunately, there are some key limitations into this that everybody needs to be. The first limitation, as I'm sure you're all screaming at the screen now, is actually there's no process measurement associated with any of that omics analysis, which is really important. So yes, we can use molecular work and we can predict who's doing stuff, but if we can't link that to process rates, stocks, and consumption, and of course production rates, it doesn't really mean so much. So it only allows you to make suggestions. What we need is tight collaboration between us all to work together to do the sorts of rate work that Jacqueline very nicely presented there, but marry it to this molecular work as well. Okay. One of the other key limitations of much of the omics work that is done is that there is normally fractionation. So unfortunately, many of us are either focused on prokaryotic organisms, which are generally very small, or they're focused on very much larger eukaryotic algae. And what they would typically do is they would sequence the DNA that they isolate from small fractions and sequence the DNA that comes from larger fractions, which is great if you just look at the one sample and you can say how abundant your organisms are and are they expressing your genes of interest. But if you want to compare the two samples together, which of course is everything that's in that marine sample, for instance, it makes it very difficult to do. Now, there are ways of getting around that. So good people around the world will put markers into their omics analysis when they do it so they can normalize between samples. And what we typically do in the study that you can see on the right is we married, so we did a seasonal study with some of the guys here, actually, with Frankie and Ruth. So we did a seasonal study of DMSP production and cycling off the coast of Plymouth in the UK at L4. So we took process rates. What you can see there is the stocks of DMS, but we did meta-transcriptomic sequencing over a 6-month period. But what we did differently to what most studies are out there, we didn't do any fractionation. So we're looking at an unbiased sample. So we're looking at the mixture between eukaryotes and prokaryotes. So what you can see here is a transcript abundance for all of the known genes that we know are involved in cleaving DMSP to make DMS, and it gives us a nice story that we can better predict which organisms and how those organisms are generating the DMS that we like to study from DMSP. So you can see here, in the majority of the cases, it's the bacterial genes that are highly transcribed and are likely responsible for the generation of DMS that you can see. But the largest spike of DMS that we see is associated with an algal, with an haptophyte abundance expressing ALMA1. So essentially the take-home message here is it's important when you're trying to do molecular work to make sure that you can do it in an unbiased way, where if you're fractionating, you're doing it in a way where you can make your fractionated samples comparable to each other. Okay. Another thing that's absolutely vital in when you're doing multi-omics, the sequences that you use and your knowledge on those sequences is absolutely vital. And this is something that we try to take to heart and try and preach to other people, but it is a very difficult thing to do. So what I mean by this is, so the way in which we will predict which organisms are driving a process in an environment from a molecular aspect is we will take the genes that we know about that are involved as marker genes for those processes, and we will look at their their abundance in a sample. The way that we do that is we take characterized protein sequences that we know have that enzymatic activity, and we make something known as a hidden Markov model. So it basically it gives an appreciation of what is known to be functional. Okay, what some people do and what you can also do is you can willy-nilly put in a lot of different proteins that you've got no information as to whether they're functional. Of course, you'll get more hits when you do that, and you will make your process seem much more important, and you'll get lots of organisms as being suggestive as being important. But what is really vital when you're doing omics, in my view, and I know other people that have similar thoughts to me, is that when you do this analysis, it's really vital that you know the limitations of the study. So you know what people understand about the functionality of the enzyme group that you're studying. So I've given you an example here of a recent publication that we put out this year in Nature Microbiology, where we identified a new enzyme involved in making DMSP via the transamination pathway. So it started off in bacteria, which you can see here in DCIG. And all of these different clades here in different algae represent a protein known as DCIE, which we made a story of because it was abundant in some pico phytoplankton. But what you can see is there are many different clades, so different phylogenetic groups of organisms, in this case algae, that contain this protein. We think it's important to actually characterize the functionality of that diversity. So we have model proteins that we can put into our HM model, which will allow us to much more accurately predict which organisms are actually driving that process or not. If you don't, then you will get an answer, but it's difficult to tell whether your answer is correct or not. And also, you of course have to set yourself a confidence level. So when those of you who are familiar with molecular biology, when you take a protein sequence and you ask if it's present in an environmental sample or an organism, you always have to set an E-value, which is the statistical value that protein is not your protein, what you're looking for. So it's really critical that you set it at the right level. And that needs to be informed by functional analysis, where people have shown, well, this protein doesn't have it, so that needs to be my cutoff. So those sort of values are detailed in the primary publications when we put these out there. And I think it's important that we put a lot of effort into understanding the functionality of these different proteins. A very good example of where it is a bit difficult to do this type of analysis is in ALMA1, for example, in the phytoplankton. There's a really cracking paper on it out there, but actually there's huge different variability in the activity of the different ALMA1 versions that are out there, and there's huge diversity in between the two functional extremes, and there just needs to be better characterization for people to be able to say, to be able to use those sort of systems to go out there and say, what is the contribution, how important is ALMA1 in environmental samples when you analyze omics data. Okay, so another thing that you can do, if you don't want to go down the route of spending lots of money and doing multi-omics sequence.
I'm sorry to interrupt you, but we are a bit behind schedule, so please, if you can wrap up and conclude.
I can certainly conclude. So essentially, what you can certainly do as well as this. Sorry, are you still okay? So you can use qPCR or RT-qPCR to check the abundance and the transcription of your gene's expression in samples, which doesn't require you to have any, to spend any money on doing omics sequence, and that can link very nicely and very easily into process measurement work when you've got the samples. What I guess the take-home message of the talk actually is make sure that we collaborate together, actually, that we do detailed sampling, we take process measurements, and that we combine it with molecular techniques, and you're considering the functionality of what we know and what we don't know. Okay, I'll stop there.